Okay, we can discuss Renesmee at a later moment. Um, the question is, what's worse, the CGI or the naming? I'm gonna go with the name. Look, this movie is so good. All right, but can I tell you, you know who is phenomenal in those movies? Kristen Stewart, who's just she's so good. She's such a good actress. Oh, no, she is. She's really good. She's like one of our finest. Especially if you've taught like a bunch of high schoolers. Like I think at the time people were like this performance is just like checked out or whatever. But like I don't know, pretty I don't know, teach a lot of high school kids. Like you're like, yeah, that's pretty accurate portrayal of high school kid. Anyway, okay. So we talked about DNA, and the first thing we actually talked about was before duplicating it, we wanted to break down that kind of idea of chromatin versus chromosome versus kind of the different ways you see DNA. And DNA, of course, is this double helix. I, of course, did a beautiful rendition of this on the board just yesterday. But if you measure across a DNA molecule, it measures about 2 nanometers across. So I went ahead and put my notes on the digital version so that you'd be able to see it. So we have these 2 anti-parallel strands of DNA, and the space between them averages about 2 nanometers. Does that make sense? If we just had the DNA like that, we said that it would be 2 meters long. That obviously isn't feasible inside of a cell that is much less than 2 meters wide. So what do? Well, the first thing is it gets wrapped around what we call the nucleosome, or it forms the nucleosome, which is this core of 8 histone molecules and the DNA wrapped around it. So we call that a nucleosome. Sorry? A histone is just it's a protein. It's a protein, and that's what it's called. And if we go to mytestymoleview.org, but not the new app, which we will note is on Notice. Oh, listen, God almighty. We don't have time for that right now. So this is a single component of histone. The whole thing, I wish we had... See, you always just... There we go. That's the basic structure of a histone. So it's just a big protein structure. But 8 of them... So you can see the histone itself is made up of 8 constituents. H2A, H2B, H3, H4. So like that's one component of it right there. And when put together, we get this, around which the DNA loops, right? And when the DNA loops around the histones, each histone has a diameter of about 11 nanometers. And when those form into a coil, we get what we call this supercoil, this first supercoiling, and that is the 30 nanometer fiber. And that 30 nanometer fiber is sort of DNA in its default position in the cell. Does that make sense? Which is already made 15 times more compact than the original DNA strand. Does that make sense? Right? Because a single strand of DNA is 2 nanometers. This is 15 times wider, but because of the curling nature of it, it uses up tons of the length. Does that make sense? So it dramatically shortens it. It's 15 times wider, and it decreases the length by on the order of hundreds of times. Does that make sense? Because of the coiling property. Coiling, very effective way to limit length. Then that coiled 30 nanometer fiber will be folded on itself. Not coiled, but folded, and that forms what we call the 300 nanometer fiber. That's an average in length. Some are wider, some are narrower. And then that itself will be coiled again, and on average, that is what we call the 700 nanometer fiber. All right, and that itself is the chromosome. And the chromosome across, because it's two 700 nanometer fibers, averages about 1400 nanometers. Does that make sense? 700 nanometers, averages out to 14, yeah. It's on there now. Yeah, I just put it on this morning. So DNA is not typically fully folded into chromosomes. The cell needs access to the DNA to use it, and it can't be fully uncoiled. It wouldn't fit. So it exists in a state where some of it is more coiled than others. And what we basically said was that genes that are under active transcription, in other words, genes that are being used in real time, will be exposed, right? And this is what we call the kind of beads on a string view, which you can't see with a light microscope, but with an electron microscope you can. Does that make sense? With a regular light microscope, that's far too fine for us to see, but with an electron microscope, which can see in much higher resolution, you can actually see this, what we call beads on a string effect, and each bead is a nucleosome. During interphase, the active chromosome, the portion of the chromosome that's being read, will be in that sort of 30 nanometer fiber. And during metaphase and during cell division, during all that, that's when you see the full 700 nanometer chromosome. Does that make sense? Cool. The study of what genes are used and when they are used is called epigenetics. All right, and then we talked about interphase and DNA replication. And then we talked about briefly pausing and saying, we know how DNA replicates. We're about to teach you. The second and the bit of this lesson that we're about to do, we're going to look at how it is that, I just want to make sure I'm actually streaming. Oh, good, I am. Okay. We're going to look at the steps that DNA goes through to be duplicated, to replicate. But keep in mind that we didn't always know that. It had to be figured out, right? So by the time that Watson and Crick had figured out the structure of the molecule, that didn't then mean that we magically knew how it duplicated. And so scientists were left in a situation where they said, well, we don't really know how it duplicates. We don't know what the mechanism is. But a good start to understanding the mechanism would be to understand, if you're making a copy of DNA, what might that look like? And they postulated that there's kind of three ideas that it could work as. One possibility would be that the original DNA from the cell gets copied, but the original strand gets left alone completely. In other words, we might read alongside it and make a copy, right? But the original one would be exactly left unchanged. Does that make sense? The second possibility would be, what if we kind of split the DNA apart? They knew from the Watson and Crick model and from Chagriff's research that about complementary pairing. So they understood one possibility is you could rip that apart and use each half as a template, in which case no DNA would be destroyed, but every new strand of DNA would be half old and half new. As it turns out, we know this is the correct one, right? But that was at the time just a theory. The third possibility is that the original DNA might get destroyed and that the new DNA might either be completely new, nothing to do with the old, or might be a random combination of the old. Does that make sense? So what they call the dispersive idea, that the DNA would not, no DNA would be intact from the original cell. And how did they test this? Well, they tested it with isotopes. They tested it by first growing bacteria in a medium where they used heavy nitrogen. And if they took that and spun it down, they would find that the DNA would collect at a certain spot in the centrifugal disk, right? Did we ever use the centrifuge before? Have you seen my centrifuge? Yeah, so you spin it down and the heavier stuff settles to the bottom. And then what they did was they transferred it to a second medium, and now they grew the bacterium, but this time in a lighter nitrogen. And if DNA replicated this way, we would expect that we would have, after replication, we would have one strand of DNA that was heavy and another strand of DNA that was light. And so we'd expect to see two bands when we spun that down. But we didn't see that. Instead, what we saw actually was one band, and it wasn't as light as pure light DNA, and it wasn't as heavy as pure heavy DNA. And so what do we conclude from that? It must be half and half. Does that make sense? Which is a fit for the semi-conservative model. The idea that when it splits, because it served as a template, this strand is half light, half heavy, and this strand is half light, half heavy. So they're both the same weight, and they're both halfway between light and heavy. And then if you let them replicate again, now you'd still have one living set of DNA that was half light, half heavy, but you'd have a new one that was all light. But you'd never get a fully heavy one again, and so that supports what we call the semi-conservative model of DNA replication. Does that make sense? And that experiment was the first indication we had of how DNA must replicate. And what we conclude from it is DNA must replicate by splitting in half, and each half serving as a template for a new strand of DNA. Does that make sense? All right. You know, on any other day, if you looked over and somebody saw somebody's face covered in blood, you'd be like, that's disturbing. I should be worried. But then there's this one day of the year where we're just like, that's cool. That looks good. No, it's a look. It's good. All right. So let's get to this. So how does this actually happen? So we're going to go through the steps. And the first step is what we call the initiation phase. So this is the mechanism of semi-conservative replication. And in phase one, so now you should have yesterday's note. If you don't have yesterday's note, make sure you mention that so you can get your copy. But where it is, God knows. I have yesterday's note right up here. I don't know what I look like. I look like a guy who doesn't have the note I'm writing. I don't know if I do have extras. Do we know who handed that out yesterday? Oh. Yeah, no, I wasn't sure I'm the problem. Not a problem. Well, I'll get those printing for you, but in the meantime, we're going to get going. So you're going to have to kind of follow along if you don't have it. Who doesn't have it? Just one. Okay. So, so the DNA has to get split. What enzyme is responsible for splitting the DNA? The enzyme helicase. Helicase, quote unquote, unzips it. Shh. Quiet. So helicase is responsible for uncoiling the DNA and then splitting it between there. We call this point the replication fork. It's like a fork in the road. Now, as the lone base pairs or the unbound base pairs are exposed, what of course is a danger here? Yeah, damage, right? Damage, mutation, because normally they're protected inside the double helix of DNA, and instead now they are exposed. And so, what attaches themselves to here? What we call SSBs, or single-stranded binding proteins. SSBs, single-stranded binding proteins. Okay, so once the SSBs have bound, sorry, I'm going to attempt to do two things at once, which is always a disaster for me. I can barely do one thing at once. Here's the note. So I just want to confirm before I print this. Only one copy? Okay, so 2 copies. Okay, so I want to make sure we know what enzymes are involved. And we've already mentioned one of them, but we'll write it again anyways. The first enzyme that's involved is what? Helicase. And what does helicase do? Sorry, it does what? Very good. It unwinds and separates the two complementary strands of DNA. All right? A second one is called topoisomerase 2. Topoisomerase 2. And topoisomerase 2 is designed to relieve stress and strain from the unwinding process. So this relieves strain and stress, relieves I before E except after C, right? Relieves. That looks right, right? Okay. As I'm sure you know, have you ever tried to unwind two like wrapped up cords and how you fix the problem here but you make it worse down the line? That ever happened to you? It sure happened to me. So the job of topoisomerase is to relieve that stress lower down. Why didn't that send? No, it is. Okay, and then we already know these, but we're going to write it again anyway. Single-stranded binding proteins. Bind to the exposed nucleotide, nitrogenous bases to protect them. And if you look here, what you'll see is that this doesn't actually happen just in one spot and work its way up the entire DNA molecule. Do you see that? It actually happens at various points throughout. So helicase is activated by a section of DNA code it's long chains of AT pairs at various points throughout each chromosome. So we often draw it like it happens just in one spot on the DNA molecule. But of course, remember that a chromosome is positively enormous, right? And so that's just sort of an oversimplification. But the actual mechanism involves that happening at various points throughout. And then these all work their way towards each other. Does that make sense? And eventually when they meet up, then the whole chromosome has replicated. Does that make sense?
Is there only 8 T pairs that activate?
Yeah. Oh, stapler is here. Hole punch should be here, but isn't. Hole punch is right there for some inexplicable reason. If you want to bring it here, that'd be great.
Is there a reason it's only 8 T pairs?
like, is there a reason? No, not really. It's just how it works. Which I know, like, in other words, you need some kind of symbol. And so throughout your genome, big sections of your genome don't code for any particular protein. Those big sections of your DNA that we long called just junk DNA. The theory for a long time running was that all of that was just useless and those genes kind of scattered throughout them. And we know big chunks of your DNA are actually viral remnants, which is interesting. Your DNA carries the history of your grandparents and great-grandparents and great-great-grandparents and all those generations going back of having viral DNA injected into the cells, absorbed, and your cells surviving that, which is interesting. So you contain vast portions of, extinct viral DNA to which we become immune. But then we've also come to understand that big sections of the DNA that don't actually code for genes do code for controlling our genes. They're part of our epigenome, where they control gene expression. And as it happens, we have sections that, however that evolved, we're not entirely sure. Did the mechanism for replication evolve, and then we started evolving those sections showing up in more places in the genome? Maybe. Thank you. I'm sorry. You're top 5 faith in you. And then, or is it the case that evolved from the fact that we already had those large sections of AT in our genome? Don't know. That's a mystery to me, and I would imagine to many. Okay, so in phase 1, we start unwinding and unzipping this, and we act to protect the single-stranded nucleotides with single-stranded binding proteins. What haven't we actually done yet? Yeah, we haven't actually made any new DNA, right? We've unzipped the old DNA to expose it, but we haven't made any new DNA. So now let's look at that. That is phase 2, what we call the elongation phase. This one here. The goal with this is just to show that this doesn't happen in only one spot. It's not like we start unzipping at the end here and unzip here and work our way that way the whole time. What actually happens is that at key spots here, 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 unzipping will start to happen. Does that make sense? And it will make its way throughout the molecule. So it's not like we start at one end of the molecule and start unzipping. We start at various points throughout and start unzipping, because keep in mind a DNA molecule is enormous. So if you started at one end and waited to get to the other, two things would happen. One, it would take a lot longer, right? And B, you would have the whole thing unzipped at once, and it would be much more likely to become a big tangled mess. Does that make sense? Whereas this is almost like it happens from the inside. Did that answer your question at all? Oh, good. Good. Okay, let's take a look. So now we have what's called the elongation phase. And the first thing is, you may remember we talked about 5' and 3'. What does that mean? Great. Does anyone remember which is which? Yeah, the phosphate group is on the 5' carbon, right? And the 3' carbon contains that free hydroxide group. But this is talking about the ends of the DNA molecule itself. And if you remember, we said that the way that DNA gets constructed on our molecule is that the 5' end of the next nucleotide gets stuck to the free 3' group of the previous. Does that make sense? In other words, DNA gets built in the 5' to 3' direction. But we also said that the original strand is anti-parallel, which means that if the 5' end of one strand is exposed, the matching side will actually be the 3' end. They're anti-parallel. They run in opposite directions, which means that when this happens, the base pairs are going to get added, and we're going to make a new chain of DNA. Does that make sense? A complementary strand. But one side of that is going to be easy to make because it naturally gets made in the 5' to 3' direction, and the DNA is getting unzipped such that we're always exposing new 3' ends to add on to. Does that make sense? In other words, see how this one, the way in which we want to build from 5' towards 3' matches the way the DNA is getting unzipped. Does that make sense? What about on this strand? the DNA is getting longer this way, but what way does it have to build? 5' to 3', which means it's going to have to start and stop and start and stop and start and stop. Does that make sense? When this much gets exposed, it can start building, but then when now it's exposed up to here, we have to go and start building again, and then these will have to be connected to each other. And then when a bit more gets exposed, we can go here and start building again, but now these two will have to be bridged to each other. Does that make sense? So we have one strand where building is easy, and we call that the leading strand. Then we have another strand where the building is going to be disjointed, and we call that the lagging strand. Does that feel okay? Does that feel comfortable? All right, you can only build new DNA by starting with a 5' end and building off of the 3' end. So you're always building in the 5' to 3' direction. If we remember, this 3' means that this was the free OH group on carbon 3 of the ribose. And that means that the first base we add is going to have a free 5' end, which, as we were told, is the free phosphate group, right? And by free, I just mean unbound, not stuck to anything. Now, we've drawn these first few in red, and the reason we've drawn these first few in red is that the first few bases added are actually RNA, not DNA. And we call them primers. Do you know what a primer is?
Like wood, you put a primer on before you put paint down.
That's right, and it helps it stick better, right? Just the nature of it, this isn't actually necessarily to make it stick better, but it's the similar idea that the first few base pairs actually get added as RNA, not as DNA, and we call them primer bases. And the enzyme that does this is called DNA primase. Annoyingly, you wish that it was called like RNA primase because what it adds is primer, but the reason we call it DNA primase is because these are priming the way to add the rest of these bases, which are going to properly be DNA. These are going to have to be replaced later. So another enzyme is going to come along after the fact and replace the RNA primers, which is just a handful of bases, usually 2 or 3 or 4, with DNA. Does that make sense? So we'll make a note of that. These will get replaced later. I'll use a drastically different color. These will get replaced later by the enzyme DNA polymerase 1. Easy to remember that DNA polymerase 1 is the polymerase that eventually will replace the first bases that got put down with DNA. Yeah, they want it's done. They are named with Roman numerals. Not sure why.
We like Roman numerals in science.
Yeah, so now we'll make a little note here and we'll say new DNA is always added by the enzyme DNA polymerase 3 in the 5' to 3' direction. That makes this strand, the leading strand, easy to write. On the other hand, the lagging strand is fragmented. What does the word fragmented mean? In pieces, that's right. So we always get these pieces here. Sorry? Well, it has to fragment because, again, writing has to be done in the 5' to 3' direction. Because, so RNA polymerase would come and start writing here, or DNA polymerase 3 rather. DNA primase would lay these primers. Then DNA polymerase 3 would come and write this. Then it's done. It's gotten to the end of where it is. So it's finished. But in the meantime, more has been exposed. So now DNA primase is going to come and lay that primer. Then RNA polymerase is going to come and add bases. And then while that's happening, it's going to get to the end. But in the meantime, more has gotten exposed. So RNA primase is going to come lay a primer here. And actually, this diagram contains an error, which is, could we have this section of blue appearing with no red there? In other words, could DNA polymerase 3 be doing what it's doing without a primer having been laid first? No. So that's a bad diagram. There should be a primer here. And specifically, this is DNA polymerase 3. All right, so both the leading strand and the lagging strand use the same enzymes. DNA polymerase 3 to add the new bases, and then DNA polymerase 1 will go in and eventually replace the primer with DNA. Does that make sense? We've already said this, but I want to say it again because it's such an important point, it bears repeating. So the leading strand has an exposed 3' end. So the new strand starts at 5'. And DNA polymerase, which you'll see written like this, DNA polymerase 3 can write the strand in one go. After DNA primase lays the primer. The lagging strand when it comes to writing a thing like this in a note, by the way, that like we've already written once, we're repeating it, that repetition is to help like cement it in our brain. An even better thing is sometimes if you're in lecture and a prof is talking about something like this and repeating something, if a prof is taking time to repeat something, it usually means it's an important concept. You often do well to try to write those sentences completely in your own words. And then we're going to repeat what we've already said, which is that what replaces the RNA primer with DNA? Nope, that lays it in the first place. What's going to replace that RNA with DNA? DNA polymerase 1. DNAP1 will replace the RNA primers with DNA. And DNA ligase seals the gaps between fragments. It will go in and it will facilitate a condensation reaction between the bases. So what happens in that gap is you would have the 5' end of one of them wouldn't be bound to the 3' end of the other. They'd just be sitting bare, and ligase will go in and cause those two to undergo a condensation reaction. So we'll say seals the gaps between fragments. You'll see the term sometimes Okazaki fragments, named after the fellow who discovered them. His name, you'll be shocked to hear, was Okazaki. So sometimes the fragments are called Okazaki fragments, just a phrase to be aware of. Yeah.
How can you tell the difference between the Roman numeral 1 and an I when you're writing it like?
You can't. So it's all context clues. So if you want to be like, is that DNA polymerase I or DNA polymerase 1? The answer would be 1, because you know from context that there's also a DNA polymerase 3, and we haven't talked about it yet, but you can probably bet there's going to be a DNA polymerase 2 if we have a 1 and a 3, right? By the way, the numbering, I'm not sure, but I wouldn't be surprised if it's just that the original one was discovered was DNA polymerase, and then we slowly discovered that they were in fact different molecules. I don't know the specific history of that, but I am always fascinated by these things in science where like the numbering doesn't match the order in which things happen. The biggest example of that being the photosystems. I always think it's comical that we say, okay, everything starts with photosystem 2. It's like, well, this does not seem like brilliant numbering, you know? You wouldn't get away with that in other things. You wouldn't be like, step 1, clean up and go home. But we all start on step 11, which is set up your, do you know what I mean? Like that would be a bad way to do things. And yet, in science, we're often like, okay, first thing, photosystem 2, followed by photosystem 1. Yeah, in that case, it's just the order in which they were discovered. Again, you have to remember, you are learning this by being like, here's what happens. When we learned this, not me, I didn't learn it, but when scientists discovered this, you pick it up piece by piece by piece. So at first we just discovered that there's these we discovered that there's pigments and they absorb light, and then we discover, oh wow, they're in a protein complex and they absorb light. And then it's only a ways down the way that they discover, oh, there's a second photocomplex. Oh, well, we better call the first one photosystem 1, we'll call the next one photosystem 2, and then it's only months and years after that that they go, oh, the actual order in which these things operate is this. Does that make sense? So you have to remember, it's all well and good when you're looking at a textbook after people have put millions of man hours into figuring something out, going like, oh yeah, that order's silly. But remember that men and women had to sit and do millions of hours of research to figure out in the order in which these things happened. And their doing so was complex. Science is a thing that is done in bits and pieces, and it is entirely collaborative. No one person figures anything out. It is always a collaborative enterprise. It's team sport. It's too big and complicated otherwise. Nobody can know everything about any individual field of science. It's just too big and complicated. People need to work together. And it's why I hate that things are given single person names, like, oh, Krebs cycle and Calvin cycle and all these things, the Bohr-Rutherford model and all these things. as if they did those things. They contributed key pieces, absolutely, but it takes millions of people to build scientific knowledge. It just can't be done by one person. It's too complicated. Yeah.
What was that one in chemistry where it's named after like a professor, but it was actually the students who did it?
Yeah. So like that's like what they always call they always call Rutherford's experiment, right? Like the gold foil experiment. And of course, he didn't do it. He helped him analyze the data. It was his lab, but it's actually the Geiger-Marsden experience. Right? I think they're the people who actually did it. But such is the way academia works. If you run the lab, your name goes up the top of the paper. It's funny because I see both sides of it, right? I remember my friend Greg was doing his PhD, and he was running all these experiments, and he used to always laugh that the top title, like the person whose name was at the top of all those papers, was always his professor who had nothing to do with them, right? Like he didn't, he would quickly proofread the papers after and go like, yeah, you're good. And of course, like Greg would run it, and then like the master's students and like whatever underneath him would do like a lot of the grunt work, and all the names were beneath them. But that's just kind of the way it's done. It's it's like everything else. Ego is as much a part of science as a part of any other human endeavor. So if any of you ever discover something, I want my name in it. You can call it the Brown Orlando postulate or whatever. Okay, so what comes next? Well, like we said, this actually happens not just in one place in the DNA, but in a bunch of places. But eventually, all of the DNA should get duplicated, right? Like if this keeps happening, if we keep splitting the DNA and we keep adding RNA primer and then DNA polymerase 3 adds new bases, then ligase comes in and connects everything up. DNA polymerase 1 comes in and replaces the primer with DNA. Eventually, you will have made 2 whole second sets. Does that make sense? And at that point, we enter what's called the termination phase. Termination phase meaning, what's it mean to terminate something? To end it, to finish it, right? Well, not necessarily. Yeah, in this case, we're not killing the DNA, but what we are doing is terminating the replication. So once the full leading strand is replicated,
Excuse this interruption. If you have Isaiah, could they please come to guidance?
Isaiah.
yep,
For Isaiah.
excellent. Thank you.
Yes.
Why does it sound like our PA system is so much quieter than every other classroom?
Right? Tell you not when Kiki's on it. I love when Kiki does the announcements. I can hear every word. Every word. All the words. Terrifying for me. That was a lot. Now, there's a little quirk about DNA polymerase, which is just the way it works. It always has to be bound on to the next nucleotide to add the current one, which means when it gets to the very last nucleotide, it can't do it. It can't add the very last nucleotide. Yeah.
This is going to be a very stupid question, probably.
Yeah.
So you know when it's like replicating and it has like it's like making a complementary strand, right?
Yep.
How do you get like
That's a great question. So the DNA polymerase 3 um, is a big protein. Oh, look at me, I'm a big protein. And here it has A, G, C, C, G, C, T. And this has binding sites for free T, Gs, and Cs, and As, and Cs, and, right? And then what it happens when the active site senses a C, this thing will undergo a conformational change that this G will come and get added to it. Does that make sense? And then it will move along the chain to the next one. And if this binds to a C, then again it will attract a G to it. So the protein of DNA polymerase 3 is designed to be able to attract all 4 free-floating nucleotides, which literally are just free-floating. They're floating around. There's machinery in the cell that's responsible for making sure there's always lots of nucleotides available in the nucleus for this to happen when replication is coming up. And then the polymerase molecule moves along the strand, and it interacts differently with a C than it does with a G, than it does with a T, than it does with an A. And it is designed that when a C is here, the G's that it has bound to will be attracted to and plant themselves there and get attached to the previous one. Did that answer your question? So it's just kind of, it's a machine that interacts with the molecule. The nice thing is that half the work is done by the fact that A and T naturally bind to each other, whereas G and T naturally don't bind to each other. But even still, the DNA polymerase 3 molecule protein is designed to facilitate that. I shouldn't say designed, has evolved to facilitate that. It functions to facilitate that. It's using anthropomorphic language again. Did that answer your question? Actually? Okay, good. I'm going to erase this drawing because that is not a drawing that's going to help anybody who doesn't know what we're talking about be like, oh, that makes sense. Okay. So there's this interesting quirk that DNA polymerase actually can't add the final nucleotide. And without remedy, this means that each generation we would lose a nucleotide off the end of our DNA, which, is that problematic potentially? Yeah. So without remedy, one base pair would be lost per generation. The solution to this is that the end of the DNA strands, the end of each chromosome, I should say the ends of each chromosome, is a junk section called the telomere. Losing a base here doesn't matter because it's junk. Right? This is non-coding. Now, even still, left to itself, after a few generations, would we have burned through the telomere? Yes. So, the telomere is completed, i.e. the missing base added later. The telomere is added back on at a later time by a different enzyme.
It's like, sorry, we had to code you and like cut off one of your fingers. We'll give it back to you later, though.
And this is the kind of stuff, it's not that this is interesting material in and of itself. Like, the telomere isn't like highly important information, but I think it's interesting because it shows that this is functionally an industrial process, you know? Where like we just, you know, this is done by machinery, and mistakes happen. And not just mistakes, but there's also just design flaws. I think of it like when you take plastic out of a mold, like there has to be the little piece of plastic on there that was like where the plastic got into the mold that like you have to cut off and sand down if you want to have a nice clean piece. Do you know what I mean? Like there's just unavoidable things that happen when you use machines to build things. And in this case, one of the unavoidable things that happens when you use a machine to build things is that the machine you use can't add that last nucleotide. So for safety, the cell makes sure that it is evolved that the ends of the DNA are junk DNA. They're not important. They're the telomere, and it doesn't matter if they get temporarily shortened, and then they can be lengthened after the fact. Does that make sense? Again, there's nothing particularly necessary about that information. I like the idea. Really quickly, I want to talk about just a couple of things that can go wrong. Though maybe we'll just push that to tomorrow, because tomorrow you're going to work on a little research project, so maybe we'll just do the first 5 minutes, we'll finish this note. We don't want to rush it, right? Is that plenty of information for is today Wednesday?
Thursday.
Today's Thursday? Holy crap, it's Thursday already.
That's what I said.
Time flies when you're having fun. That's right? And tomorrow's November.